Hello and welcome to the 4th UK NEQAS for Microbiology webinar on new drugs for new bugs, delivered today by Dr. Mandy Wotton.
Okay, so I'm going to talk about new drugs for new bugs. I'm going to cover some of the drugs that you may be testing in the near future and not the really new drugs that are coming through. But why do we need new drugs? Well, actually, after lots of lobbying and about a decade of people like me banging on about antimicrobial resistance, quite a few healthcare agencies, including BSAC and Public Health England in this country, and also the WHO, have developed plans to combat AMR. In 2011, I was with a BSAC group who went to Westminster to highlight the increasing problem of AMR. We had a lovely day in the Houses of Parliament, and we spoke to lots of politicians who were very interested in the subject. I did manage to get a Houses of Parliament pencil, which I still have to this day. But more importantly, it provided the impetus for Jim O'Neill, who was a politician back then, to set out plans to combat AMR in a 2014 report. He then went about and did another report in 2016 calling for a little bit more action. So one of the things he was calling for was stewardship of antibiotics. And at the moment, we've got a very good stewardship tool called AWARE. And so this stands, the AWARE bit stands for, the A stands for access. So these are the access antibiotics. And these antibiotics are commonly used antibiotics, first and second line choices. And they have a particularly low potential for resistance. The W stands for watch. So these are a group of agents which have a higher potential for resistance or are prone to selection. And they should be targets for stewardship. So you shouldn't really be using these a great deal. And then the R, the reserve agents, are ones which we really should be reserved until use. So this is quite a nice stewardship tool for clinicians to use. And this is why we've got to this point, really, because this is a scale from the 1900s to the 2000s. And as you can see, throughout the ages, we have developed and discovered lots and lots of new antibiotics. And then we hit sort of late 1990s, and nothing new came on board. And a lot of the pharmacy or pharmaceutical industry actually shut down their antibiotic units. So because it just wasn't viable financially. And again, this is the reason why. So this shows the amount of potential antibiotics that could be researched here. And then this is how long it takes in years to get to one that's actually put on the market. And this is the cost. So you can see that, you know, they have to go through about 7 to 10 years before an antibiotic is put on the market. And a lot of money is spent. And quite often with antibiotics, you know, you get it to this point and it will be put on the red list to reserve. And so you won't really get your money back. So many governments and charitable initiatives have helped to develop drugs by providing funding and so forth. But even with that, the WHO published a report earlier this year, and it suggested that only 43 antibiotics are in the pipeline, and none really focus on MDR Gram-negatives like carbapenem-resistant Acinetobacter baumannii or Pseudomonas. They also found that there aren't many oral use antibiotics on this list. So we're still in a bit of a position where we do need new antibiotics. Why do we need antibiotics? Well, I'm sure you've seen these before. The prevalence of carbapenem-resistant bacteria is still on the rise. And here we see the heat maps from the EASnet surveillance data. I'm sure you've seen these before, but these are heat maps. So the bluer or the cooler the color, the lower the resistance rate, and the redder or hotter, the higher the resistance rate. And here we show E. coli, carbapenem resistance in Europe, and you can see that for the majority of the UK and for Europe, it is less than 1%. For Clebpneumo, very, very different story. Okay, Northern Europe and Scandinavia is less than 1%, but as soon as you get down to the Southern European countries, we're talking about 25 to 50% in places like Italy, and over 50% in Greece. Pseudomonas again is just exactly the same. You know, as soon as you get down to the southern countries, you start to see this prevalence rise. If we go to the far right corner, you can see here, this is Acinetobacter, carbapenem-resistant Acinetobacter, and throughout the Southern European countries. So things like Greece and Italy and Spain, they're all greater than 50%. So this is really concerning, and we do need new drugs to tackle this issue. And of course, the resistant bacteria have a cost to the patients. This paper used the EARSnet data from 2015 to look at the attributable deaths, and also the disability adjusted life years or dailies of patients with AMR bacteria in Europe. The median number of infections was over 671,000, with an estimated 33,000 deaths. So this is not a trivial issue. And the data here, the diagram shows the dailies. So the bigger the spot, the bigger the impact. And up here, we've got third generation Cephalosporin E. coli, then MRSA. And then down here, we've got 3GC resistant or third generation Cephalosporin resistant Clebpneumo. And then in the box, we have carbapenem-resistant Acinetobacter, carbapenem-resistant Clebpneumo, and even colistin-resistant Clebpneumo. So you can see there's a lot of gram-negatives that have an impact on patients' well-being, patients' mortality. And this is from a meta-analysis literature review estimating the burden of AMR. So this is a meta-analysis, and that it looks at all of the data that's out there, all the studies that is out there, and puts all the data together. And it gives us an odds of mortality happening when a patient has a variety of different resistant bacteria. The further to the right, the higher the odds of mortality happening. And you can see where I put the lines here. You know, for XDR Acinetobacter baumannii, so very resistant Acinetobacter, KPC positive Clebpneumo, the odds are really quite high. So again, you know, this is something that we need to find drugs for. So what I'm going to do today is to go through the different aspects of the new agents I'm going to cover, and I'm only going to cover 3 in detail. So I'll cover the mode of action, spectrum of activity, a brief mention on the licensed indications, and then how to test susceptibility testing if there are any problems or anything, any notes that you need to take note of. And then I'll cover the resistance as well. So, and binding proteins in the bacterial cell wall. And it does that by, with regards to the amine group here. So that's how it binds to the penicillin binding protein. And of course, once it's bound to the penicillin binding protein, it will cause lysis of the cell wall and eventually cell death. So this is a bactericidal drug. The hydroxyl group up here, this is what makes it active against Pseudomonas and also active against ESBL, against non-ESBL class A enzymes. So things like TEM1 and TEM2 and SHIV1, some OXAs, some AMP-Cs, but just as we well know, ESBLs hydrolyze ceftazidime by binding to that hydroxyl group. Avibactam is a new non-beta-lactam beta-lactamase, so it doesn't have a beta-lactam ring. And it binds to the hydroxyl group of the ceftazidime to prevent the binding of the beta-lactamase and thus any inactivation by beta-lactamase. It collects in the periplasmic space, as does ceftazidime. And so essentially what it does is to restore the activity, the ceftazidime activity to the ESBLs. So it's quite a nice combination. The mode of action. Oops. So we've done the mode of action, spectrum of activity. This is, data comes from a very nice report from David Livermore et al. And shows the activity of Cas-Avi for carbapenemase producing Enterobacterialis and Pseudomonas isolates in the UK. And they took the first year of Cas-Avi being available and tested all of their isolates, which was 2015 now. And they showed that 95 to 99% of Enterobacterialis with KPC, GES, or other class A carbapenemases, plus those with OXA48-like carbapenemases, those with ESBLs and those with AMP-C enzymes, were all susceptible to Cas-Avi. So this is really quite special, given the numbers that we saw earlier on. They also found some Enterobacteriales where it was ceftazidime resistant, but they couldn't really find a mechanism. And 87% of those were still susceptible. So that's really nice. For Pseudomonas aeruginosa, the majority were carbapenem resistant, but due to not carbapenemases, but de-repressed AMP-C. But it didn't, Cas-Avi didn't have activity for those with efflux-mediated resistance. So in a typical AMP-C porin loss carbapenem-resistant Pseudomonas, you'd have high imipenem MICs and lowish meropenem MICs. But for those with efflux mechanisms in, you'd have high carbapenem MICs to both carbapenems, IMI and mero. So those weren't affected so much. Obviously, all of the isolates with the metallo-beta-lactamase were resistant to Cas-Avi because it doesn't have activity against metallo-beta-lactamases. So this is your NDMs and your IMPs and your VIMs. In the same vein, most K1 producing K-oxytoca producers were resistant, and also those Acinetobacter baumannii with oxygenes were also resistant. So we can see roughly where the activity lies. And this is table 2 from that study. At the top, you can see that these are the class A, so KPC organisms, KPC producing organisms and GES organisms. And you can see that on the top, you've got MIC distribution data for ceftazidime, and then the line below is ceftazidime with avibactam. And you can see the shift in MIC. So for KPC producers, most of the CAS MICs are up here around 64, whereas if you add avibactam, they're down here near to about 0.5 or 1. For OXA48 producers, you can see a similar kind of story. There is more variety with OXA48 producers because of the expression levels, but even so, you can see that drop as the avibactam has activity against the enzymes. So these are the metallo-beta-lactamases, and you can see that quite often you do get a slight drop, but not enough to be effective treatment. So most of the MBLs aren't affected by Cas-Avi. And then just below we have some co-producers. So these are organisms which have 2 carbapenemases, as if one isn't enough, some do have 2. And you can see obviously the obvious drop when you have a KPC and OXA48, but not when another carbapenemase is co-produced with a metallo-beta-lactamase. License indications. So this is from the NICE website, and at the moment in the UK, Ceftazidime is recommended for the following indications. So complicated intra-abdominal infections, complicated UTI, including pyelonephritis, HAP and BAF, plus infections caused by gram-negative bacteria with limited treatment options. And it's probably this latter one that the majority will be used for in the UK. This is a very nice 2-page summary of all of the evidence that NICE has collected to review the use of Ceftazidime in the UK. So it's really quite nice for clinicians to get hold of that. With regards to susceptibility testing, well, the easiest way to do it is by EUCAST disc diffusion method. This is a 0.5 McFarland onto Muller-Hinton using a disc with 10 micrograms of ceftazidime and 4 micrograms of avibactam. You incubate aerobically for 16 to 20 hours at 35 degrees C. So this is pretty standard methodology we're using here. One of the differences that don't forget, this is an inhibitor enzyme, inhibitor drug that we are testing here. So we'll need 2 QC organisms, a susceptible E. coli, if it's Antibacterialis you're testing, or a susceptible Pseudomonas, if you're testing a Pseudomonas. Plus you will need this Klebpneumo ATCC 700-603, which is an ESBL producer. And it's this organism which will check your inhibitor section of the drugs. It is interesting to note also that this 700-603 does have 2 colony types, and that's normal. So don't be worried when you have 2 colony types with this organism. Just use both colony morphology types. They're both the same thing. It's just a little bit odd. Below you can see the ranges for the QC, and here we can see the ranges from the EUCAST development lab. The way that they set these is by sending the organisms out to lots of different labs in Europe, and when the results come back, they combine them all to so many observations, and they then these distributions are then are the basis for the range, the zone diameter range. And you can see they're all pretty tight ranges, so there isn't an issue with QC. When you're reading the plates, you should read from the back, which is normal. And you look for complete inhibition. Don't forget, this is a bactericidal drug. In most cases, you will get a graduated zone edge. So you have to be slightly careful when reading, and I'll talk a little bit about this when we go through the MIC strips. For the EUCAST disc diffusion method, we have breakpoints for all Antibacteriales. The MIC breakpoint is 8 and 8, so that means if you have an MIC of 8 or below, it's susceptible, and anything above 8 is resistant. And a disc diffusion zone diameter breakpoint of 13, so 13 or more is susceptible, and anything below 13 is resistant. On the right are the EUCAST data for zone diameter distributions for Ceftazidime. And if you haven't seen these before, then your zone diameter is across the bottom here. Number of observations is up the top. And then the colors denote the MIC. The cooler colors, the green colors, they're the susceptible isolates, and the red and orange colors denote resistance. The dotted line here is the zone diameter breakpoint, and you can see that roughly all of the isolates colored green are to the right, i.e. give you big or larger zone diameters, and all of the orange and red are below the line, which means that this is a pretty good test for splitting out those isolates that are susceptible and those are resistant. There's a little bit of crossover, but quite often you never get these perfect. For Pseudomonas, your MIC breakpoint is 8 and 8, and your disc diffusion is 17 millimeters. But this time there's an area of technical uncertainty. So if you again look at this table here, you can see that the red box denotes the area of technical uncertainty. And if you can see here, you've got quite a significant number of isolates with MICs of 16 milligrams per litre. And, you know, I don't think in this particular case, the distaffusion method will accurately split the phenotypes of susceptible and resistant without this ATU. How do we resolve that? Well, we will almost certainly perform an MIC. And the MIC breakpoints don't have an ATU, so you can resolve it pretty easily. The other guidance for dealing with an ATU is to report as resistant or suppress a result and add a comment to say that you're unable to have a patient who is hard to treat. So I don't think that's a viable option. There aren't any breakpoints for Acinetobacter baumannii, so we won't discuss those. So gradient strips. So MIC methods are okay for Cas-Avi. Most labs will find the gradient strip convenient. These have only been validated for Enterobacterialis and Pseudomonas aeruginosa. And the manufacturer's instructions are the same for EUCAST distaffusion. So Muller-Hinton agar, 0.5 McFarland, 35 degrees C, and 16 to 28 hours incubation. The same QC organisms are used, and you read from the front of the plate, obviously with the gradient strips, and look for complete inhibition. And so above here, the Pseudomonas aeruginosa at the bottom is slightly more different. It's more difficult to read because it's got this graduated edge here. And you read at full inhibition. So the MIC would be about 6. So it's usually best to test both ceftazidime and Cas-Avi when you're reporting. But you should be aware that sometimes you really sort of need to take a bit of a look at your results before you report them out, because sometimes they can be misleading. So if you've got a ceftazidime result of susceptible and a Cas-Avi result of susceptible, no problem. If you've got a Cas-Avi result of susceptible and a Cas result of resistant, no problem, especially when your Cas is greater than 32 and your Cas-Avi is 0.5. It just means you've got an ESBL in there or something, or a KPC, and you have got lovely activity. However, this is a Pseudomonas down here, and we've got MICs to both ceftazidime and Ceftazidime-avibactam of 2. Now, these are the breakpoints here. Even if this was an Enterobacterialis, it would still be I, susceptible increased exposure. And even for Pseudomonas aeruginosa, it would be I, susceptible increased exposure, because of this off-scale breakpoint here. However, if we wanted to report that, you'd really, if you just released it and you didn't release the 2 and the 2, if you just released those 2, the medic would probably be tempted to use the Cas-Avi, when actually the Cas will do. Not realizing that they probably don't need to use the Cas-Avi, they probably can get away with using the ceftazidime. But if you then go to the dosages tab, you will see that if you get an I result, it means that you have to use the higher dose. So you would be using 2 grams times 3 IV, or 1 gram times 6 IV. And the Cas-Avi, if you use that as a standard dose, well, actually there is no standard dose. There is only one dose. There's no standard or high. And hey presto, it's 2 grams ceftazidime and 0.5 avibactam. So you can see that the high dose is the same. So I wouldn't release this report without a comment to say that the Cas-Avi is having no effect and ceftazidime should be used. So ceftazidime would be just as good in this organism as Cas-Avi. And of course, ceftazidime is probably cheaper. So resistance rates, resistance is low for now, as you would expect. When a study looked at the MDR Enterobacterialis, was less than 1.6% which were resistant to Cas-Avi, and 0.6 when you included all the susceptible ones as well. So very few were susceptible. For Pseudomonas, the rates is about 4 to 8%. Again, quite low. I suspect that there will be, as it gets used, there'll be more resistance coming through, and that will make it easier to determine what the resistance mechanisms are. So I'm going to say what the three main resistance mechanisms to Cas-Avi, but quite honestly, these are all associations at the moment. So the most common one found is a mutation in the delta region of KPC, carbaphenemases, but it's also been found in CTXM and VEB genes. And this delta region is where the carbaphenemases bind to the beta-lactam. This was the first proved in a lab, but actually later on, patients' isolates were found to have these mutations as well. Some of these mutations cause increased ceftazidime hydrolysis by trapping the drug and preventing the avibactam binding. So this is roughly how it works. Resistance has been induced in OXA48 isolates in vitro, but not in clinical isolates yet. Next is the chemical modification or expression changes. AMP-C enzymes in Pseudomonas aeruginosa isolates from CF patients have been shown to be de-repressed, which means that the AMP-D, the regulator for AMP-C, gets mutated, and that causes AMP-C expression differential. You can also get mutations in the delta loop of the plasmid AMP-Cs in Cleb pneumo, and that can cause increased ceftazidime hydrolysis as well. Finally, there's permeability changes, porin mutations and efflux mutations, but these very rarely cause resistance on their own. They're usually combined with other mechanisms. And so we can clearly say that Cas-Avi does not have activity for metallobeta-lactamase producers, but however, aztreonam is a monobactam which is not really hydrolyzed that well by metallobeta-lactamases, usually due to its poor binding. However, most metallobeta-lactamases have ESBLs and AMP-Cs alongside their MBL, and if aztreonam is linked to avibactam, then the avibactam can knock out the ESBLs and AMP-Cs, and then that allows some activity of the aztreonam to attack the metallobeta-lactamases. And sometimes we've done some synergy testing in our reference lab to look at how effective this might be in vivo. So this is an E. coli up the top here. You can see very high MICs to imimeru, Cas-Avi, and aztreonam. And then when you put them together, so this is a way of looking at synergy testing, you can see that synergy is happening between the two. So the avibactam is knocking out the CTXM, and the aztreonam is then having an effect on the NDM. Same happening with Pseudomonas as well, so very high MICs for Pseudomonas, but the avibactam is knocking out the AMP-C, so the VIM can be attacked by the aztreonam. Now, it's very important to say, that synergy testing using gradient strip is a non-standardized method, and it shouldn't really be used very easily. But sometimes if medics are looking for a little bit of comfort, with regards to a very difficult patient, then this can be done. And it is also important to say that dosing using Cazavi and astrienam in the patient, it can be difficult as well. And currently this isn't licensed, and some trials are going on. So that's Cazavi. I'm now going to start on Ceftolozane-tazobactam. Ceftolozane is a 5th generation cephalosporin with a similar structure to ceftazidime, so it disrupts the cell wall. There's a big difference is that it has a modified side chain which gives it potent anti-pseudomonal activity. And even they are resistant, even when they're resistant to carbapenem, so due to AMP-C and porin loss and efflux. It's similar again to ceftazidime in it that has activity against the non-ESBL beta-lactamases such as TEM1 and SHIB1, but obviously ESBLs easily hydrolyze Ceftolozane-tazobactam. Tazo is, we know Tazo from its combination with piperacillin. It's a beta-lactamase, the beta-lactam ring is there, and it binds irreversibly to the active site of the beta-lactamases and forces chemical changes in the enzyme, which inactivate it. And this then restores the activity to ceftolozane. Again, it's worth saying at this moment that some supplies of Ceftolozane-tazobactam have been interrupted due to a contamination issue in the production site. So unfortunately, I don't think any hospitals in the UK have access to this drug to use at the moment. I'm sure that will get sorted out at the moment, and I'm sure we'll be testing it pretty soon. Spectrum of activity. Pseudomonas species, especially the multi-drug resistant ones with resistance to carbapenems, ceftazidime and piperacillin-tazobactam. It's useful against Pseudomonas with the SBLs and de-repressed AMP-Cs and porin loss and efflux. So those are the ones that can be carbapenem resistant, but not to the carbapenem resistant isolates with a metallo-beta-lactamase like VIM or KPCs for that matter. It's active against DSBLs and AMP-Cs producing Enterobacterialis, providing they are non-carbapenemase producers. It's not active against Acinetobacter species. And in this study of Western European isolates between 2012 and 2011, we can see that for Pseudomonas aeruginosa, most of the MICs were around 0.5. Most of the meropenem non-susceptible Pseudomonas aeruginosa were around 0.51, and ceftazidime resistant ones were between 1 and 4, all of which are well under the breakpoint, which is here, which by the green bar. For Enterobacterialis, again, you can see that the majority of isolates are below the breakpoint of 2 here. So really nicely susceptible. It does have activity against Haemophilus influenzae, even those with a beta-lactamase. And this Sentry study, the Sentry surveillance program, is a worldwide surveillance. It looked at 900 Haemophilus influenzae. Some were beta-lactamase susceptible, some were beta-lactamase positive. But it doesn't matter about the beta-lactamase because about 98 to 99% were susceptible. And these come from all kinds of infection types. So that's quite a nice drug for Haemophilus influenzae. It also has activity against anaerobic bacteria, mainly against B-Frag, but limited against other Bacteroides species, as you can see here. I'm not going to spend too much time on the anaerobes, but it has activity against a range of others as well. The MIC50, so the MIC50 is the MIC which inhibits 50% of the population, and the MIC90 is the MIC which inhibits 90% of the population. So you can see they're quite low for B-Frag and for some of the others, so quite nice. The licensed indications are very similar to Cazavi, complicated intra-abdominal infections, complicated UTI with pyelonephritis and HAP and VAP associated pneumonia. For susceptibility testing, pretty much very similar to your normal standard EUCAST diffusion for Pseudo and Enterobacterialis, and same for Haemophilus influenzae, so Muller Hinton F and CO2 for the Haemophilus. Again, because it's an inhibitor we've got here, we need to add the two QC isolates, so a susceptible one and also this ESBL producing Kleb pneumo here. And then for the Haemophilus influenzae, the same story. So add this Kleb pneumo. The distribution should be read from the back of the plate if it's Muller Hinton A, and from the front if it's Muller Hinton F. And again, you are looking for complete inhibition. Just be aware that with Haemophilus influenzae and beta-lactam agents, you might get this hazy growth inside, which occurs if the plates are too wet. And the way to get rid of that is to open your, the bags that the plates come in, and let the plates dry sufficiently before you put up your test. There are breakpoints for all Enterobacterialis. The MIC breakpoint is 2 and 2. So an MIC of 2 or below is susceptible, and anything above 2 is resistant. And the diffusion is 22. But we do have an ATU of 19 to 21. So again, how do we resolve this? We can perform an MIC, because the MIC will give you a definitive result because it's got no ATU, or we can report as R. As I said before, I don't think that's a reasonable thing in this particular circumstance. For Pseudomonas, the breakpoint is 4 and 4, and the diffusion breakpoint is 23, 23, and no ATU. And then for Haemophilus influenzae, we have a breakpoint of 0.5 and a diffusion breakpoint of 23. But there is an ATU, but just be aware that if you have a zone diameter within this ATU, it only really applies if you found resistance mechanisms by it testing positive on your Pen 1 disc. Most labs will do a gradient strip. These have been validated for Enterobacterialis and Pseudomonas aeruginosa, so we can't use them for Haemophilus at all in this particular case. And with all gradient strips, you use the manufacturer's instructions to put up. In this particular case, it is the same as your EUCAST disc diffusion, so Muller Hinton agar and 0.5 McFarlane and so forth. The same QC organisms. You read from the front of the plate this time, and again, looking for 100% inhibition. These pictures show how to read the gradient strips. Pretty straightforward. There isn't really a reading issue with Ceftolozane-tazobactam. It's fairly straightforward. But again, it's early days with this testing, and if problems arise, then they usually occur when people start to test in earnest. For resistance mechanisms, pretty low again, 7.8% of Pseudomonas from a recent Spanish study were resistant to Ceftolozane-tazobactam, and a study in the UK found that just under 90% were susceptible to Pseudomonas. 90% of the Pseudomonas aeruginosa were susceptible to Ceftolozane-tazobactam. The resistance mechanisms aren't really fully elucidated, as with Cas-Avi, but there have been some odd things that have been associated. So structural changes or overexpression of AMP-C, and cross-resistance with Cas-Avi, things like mutations in an oxygene in the Pseudomonas aeruginosa, and even some PBP3 mutations have been associated with them. So now we come to Cefiderocol, and this drug has caused quite a lot of excitement recently because it has activity against metallobeta-lactamases. More on that a little bit later. So the commercial name for this drug is Ceftolozane, and really it's a cephalosporin, and you can see up the top there that it's a cephalosporin with an iron binding or siderophore side chain. And these siderophores bind iron, as the name suggests, and that's how they get into the cell. They get into the cell using the bacterial siderophores, and they get in to the periplasmic space where they bind to the PBPs and stop cell wall synthesis. So these are bactericidal drugs. The spectrum activity is pretty large, to be quite honest. It covers lots of Enterobacteriales, the Proteaceae, Morganiaceae, Acinetobacter baumannii, Pseudomonas aeruginosa, and then some of the more hard to treat organisms such as Achromobacter, Stenotrophomonas multifilia, and Burkholderia capacia complex. It also covers quite a few resistance mechanisms. So, for example, ESBLs, Cs, the class A carbaphenamases, but also OXA48, the class Ds, and the class Bs, metallobutyllactamases, IMP and NDM and Enterobacteriales. It'll cover even OXA23 and Acinetobacter baumannii, and the metallobutyllactamase VIM in Pseudomonas aeruginosa, and it also has some activity against the Cleb pneumos and the Pseudomonas aeruginosas, which are carbapenem resistant, but without a carbapenemase. And in a study of multi-drug resistant gram-negative bacteria, this is a worldwide collection, and you can see the range on the right here of different organisms and different resistance mechanisms. And here you can see the MIC50. So this is the MIC by which 50% of the population is inhibited. And you can see that even for E. coli and Pseudomonas and Acinetobacter baumannii, the MIC50 is around 0.12 to 0.5. And the MIC90, so this is the MIC by which 90% of the population is inhibited, is around 2 or 4. And even when these organisms have all the most common carbapenemases and also colistin resistance, you can see the MIC for MIC90 doesn't really go up that much, if at all. It's around 2 and 4. And that's great when you compare it against the EUCAST breakpoints down the bottom of the slide, which is 2 for Enterobacteriales, Pseudomonas, and the PKPD breakpoint is 2. So you can see that the majority of these isolates are actually sensitive. Achromobacter species are common in CF patients. They have lots of intrinsic resistance. There aren't any breakpoints at the moment, and there aren't that many large studies to show you. But actually, it has been used in salvage therapy with patients with these infections. And the MICs in my lab, just on a handful of CF isolates, are around 0.25 to 0.5. So they're fairly low. So that's very encouraging. And the same can be said for Burkholderia capacia complex, another CF organism, lots of intrinsic resistance. And in a 2016 multinational study, the MIC90 was 0.03. Similarly, with Stenotrophomonas multifilia, another difficult drug, the MIC90 in the same study was 0.25. And again, in our lab, the table on the right shows that the MICs are roughly in the same area, so 0.25, 0.12. This drug's also had said to have had activity against lots of unusual bacteria that we don't tend to see in normal patients, but we do see in cancer patients. And it's quite encouraging that it has activity in this area, because they're usually very difficult to treat. So those are the UK and US licensed indications at the moment. I'm not going to say too much about those. As for susceptibility testing, well, generally MIC determination by broth microdilution is the gold standard. And this is a general susceptibility testing broth microdilution panel, whereby you have growth control on the right here, and then along the rows you'll get growth and doubling dilutions of the antibiotic, and where the inhibition takes place is the MIC. So in row 1, it's 1 milligram per litre, and then in row 2 and 3, it's 2 milligrams per litre. So you get the growth and then the no growth. So susceptibility testing of Cefiderocol, you just have to remember that this is a siderophore. So it binds iron, and there's lots of iron in Muller-Hinton broth, and it's there for a reason. It helps the growth of bacteria. At the moment, susceptibility testing is slightly limited in the fact that there may be 1 or 2 freeze-dried panels available in the UK. These don't use iron-depleted Muller-Hinton broth, which is what you should use when you're doing a broth microdilution test in-house. It uses its own cation adjusted Muller-Hinton broth, and it uses an inoculum of 1 × 10⁵ colony forming units per mil. You seal the freeze-dried panel with once you've inoculated it with a sticky covering, and then you stack it three plates high in an incubator at 35 degrees for 18 to 24 hours. You use the usual QC organisms, and then they have a reading guide here, which as you can see from the top left is just, you don't actually see the panels, you just see a diagram. And basically they're saying that you should check the growth control first, which is standard practice, and then turbidity or a button indicates growth. For Pseudomonas, you should read at 80% inhibition, and for other organisms, you read at 100% inhibition. And basically on the top right diagram, you can see that the first row looks like a normal MIC, whereas the first growth where you haven't got growth is the MIC. And row 2 shows a skipped well where you've got no growth, but then growth in the well after it, and the red ring indicates where you should read the MIC. And then the next 2 rows indicate where you have contamination on the right with the dot with the yellow fuzz around it. And the bottom 2, again, where there's contamination and you cannot read an MIC, so you have to perform it again. I don't find this particularly useful because sometimes in broth microdilution, it's very difficult to determine what is contamination and what is your organism. When EUCAST did the testing for development of the DISC method, they used the 2 different panels. So they used the panels that we've just been talking about, but they also used a reference frozen panel set using the iron deficient Muller-Hinton broth. And they get slightly different results. One of the reasons is because the inoculum is different. So the freeze-dried panels we talked about before uses a 1 × 10⁵ CFU per mil. And the frozen reference panels using the iron deficient broth use 5 × 10⁵ CFU per mil. And if you look in the international standard for broth microdilution, the inoculum should actually be 5 × 10⁵. So EUCAST are very clear on what the inoculum should be and have done this testing for decades now. And, you know, this is always the inoculum that you use. But obviously, this is a kit that we're using with a sensor titer, and they have done their own validation. So generally, the freeze-dried panels may give a slightly lower MIC, but over 90% are within 1 log dilution for Enterobacterialis, Pseudomonas, and Acinetobacter baumannii. So not too much difference. Just be aware that there will be a slight difference, and it will be slightly lower. So the EUCAST also give in their guidance some very, very nice explanations of where you should read these panels. So they're saying that you should read the panel where there's the biggest drop off. So you do get some trailing endpoints. So on the picture at the lower end of the slide, you can see row 1, the red ring denotes where you should read it. And you can still see a very tiny button there, but they're saying that if the button is less than 1 millimeter, or there's a presence of a light haze or faint turbidity, you should ignore that and you should read where the greatest drop off occurs. And the same in the second row, you can see a very, very slight haze at the bottom of the well, but that's where you should read it. And on the bottom row, again, you can see a slight button in well 9, but that is where you read the MIC. You can test by gradient strips. At the moment, there is only 1 manufacturer which is validated. The other manufacturer, the validation studies will be ongoing this year, so should be out at the end of the year. Just be aware that these will only be validated for use with Pseudomonas aeruginosa, so you cannot use them with Enterobacterialis. And you will note that you use the standard Muller-Hinton agar, not the iron deficient. The reason for that is with most diffusion methods, including the disc diffusion, people think that the iron is actually caught up and bound in the agar, so there's no need for iron depleted agar, because the diffusion, it doesn't affect the diffusion of the drug. You use the standard Pseudomonas aeruginosa QC strain, and this is a bactericidal drug, so you should read at full inhibition, which includes hazy growth and colonies within 3 millimeters of the strip. And on the pictures to the right, the one below, you will see there's lots of little colonies floating around the bottom of the ellipse, and you count all of those little colonies. Right at the bottom of the slide is a table which shows that the categorical agreement is over 99% for Pseudomonas aeruginosa. Just bear in mind that this is printed in the package insert of the manufacturer's strips. For EUCAST, there are MIC breakpoints with no ATU, and there are 2 for Enterobacterialis and 2 for Pseudomonas aeruginosa. For Cenotrophomonas multifilior and Acinetobacter, there is IE in the tables, which mean insufficient evidence, but you can use the PK/PD breakpoints where susceptible equals or less than 2 mg per litre. So very useful to know. Disc diffusion methods, again, these were validated with reference, frozen reference BMD panels, and we can perform them on Enterobacterialis and Pseudomonas aeruginosa, so not all Pseudomonas. Cenotrophomonas multifilior and Acinetobacter, there are no clinical breakpoints, but you can use the PK/PD breakpoints. And again, you use a normal Muller-Hinton and the normal conditions for a EUCAST disc diffusion method with the usual QCs. So here are the breakpoints below, and for Enterobacterialis and for Pseudomonas aeruginosa, you will see that they are the same, but they both have an ATU, an area of technical uncertainty, which is slightly different for Enterobacterialis and Pseudomonas aeruginosa. And right at the bottom of the screen, you will see that actually in the notes section in the EUCAST tables, you will see for Cenotrophomonas and for Acinetobacter baumannii that you can use a disc and you can read according to 20 millimeters and 17 millimeters, and they both correspond to the PK/PD breakpoint. I'm pretty sure that eventually these will be added into the table as proper breakpoints for disc diffusion. And this is just to demonstrate to you the reasons why we have areas of technical uncertainty. So top left, we have E. coli, top right, we have Kleb pneumo, and these are classic EUCAST histograms where if you had the breakpoint in the middle, you should see all of the green colours on the right, which denote susceptibility, and all the red and orange colours on the left, which denote resistance. And you can see with the boxes, the highlighted boxes, the ATU boxes, that you get a multitude of MICs within those boxes, which means that in that area, in those millimeters, those few millimeters, we can't decide whether it's susceptible or it's resistant. So it is an area of technical uncertainty. We can't be sure of that phenotype. There, on the bottom right, you can see it for Acinetobacter baumannii and Cenotrophomonas, and you'll see it's a little bit clearer on those histograms. But to be honest, more testing needs to be done with those, more isolates need to be gathered. So how do we deal with the area of technical uncertainty? Well, we can resolve by just performing an MIC, because the MIC breakpoints, there is no area of technical uncertainty. Or we could report as resistant, and sometimes with other drugs, you can report as resistant. In this case, I'm not so sure with this drug. So in summary, there are some issues with MIC determination. Reading may be difficult at times due to the trailing endpoints. And there is an ATU to deal with in the disc diffusion, but Generally, the reproducibility of broth microdilution, the gradient strips and distribution is quite good, given the limitations. And quite often, we're going to use this drug in patients with limited treatment options. So I don't think it's useful to report anything with a measurement in the ATU as resistant. I think we want to resolve that by doing an MIC. And I think at times with this drug also, because of the indications it's used for, this drug's going to be used prior to AST only in certain circumstances, and the drug may be used on clinical signs and not on AST use. Resistance is fairly low at the moment. There have been quite a few things associated with resistance development, including mutations in various enzymes, carbapenemases, and also mutations in the ion transport genes. But I think a lot of work needs to be done on this a little bit more, and we don't quite know what the resistance mechanisms are in some of the agents, some of the organisms so far. So I think we need to wait before it's being used a lot more than currently. Some of the other new agents on there is imipenem-relebactam, which has activity against Enterobacteriales, except Morganellacea, Pseudomonas aeruginosa, and Acinetobacter, and it's active against ESBL, XAMPC, KPC, but not metallo-beta-lactamase and oxacillinases. Meropenem-avibactam, active against all Enterobacteriales, Pseudomonas aeruginosa, but not Acinetobacter, and active against the ESBL, XAMPC, and the Class A carbapenemase KPC, but not MBL and oxacillinases. And then finally, a tetracycline type of agent, ravacycline. It's active against all Enterobacteriales, Acinetobacter, but not Pseudomonas this time. And again, it's active against ESBLs, KPC, and carbapenem-resistant Acinetobacter, which is conferred by a metallo-beta-lactamase or an oxacillinase. So as you can see, there are new hopes after having virtually no new gram-negative agents for over a decade. We are on the cusp of having some new agents to put in our armory against the MDR organisms. Thank you very much for listening.
Thanks very much, Mandy, for that really interesting talk. I think I've certainly learned a few things from listening to you today. So I've got some questions then from our audience. I'll start you off with a few easy ones. So at the beginning of your talk, you had a list of antibiotics that were coded in green, yellow, and red. And we've got an anonymous attendee has asked, will the red list of antibiotics be available afterwards, or are they available somewhere?
Not quite sure of what that means, but they're all part of the AWARE campaign. And I think if the attendee meant, are they available currently to use, then they need to go to their local formulary and local pharmacy to know that, a pharmacist or the antibiotic pharmacist that's in their hospital to know that. If they are asking, is that, can you see that the AWARE campaign and what drugs are on what list available, then yes, I'm sure the slides from today are going to be available, but also just type AWARE campaign into a search engine and you will see them.
Okay.
Hopefully I've answered that question.
I think you've covered both ways of interpreting it, so that sounds good. We have another question from an anonymous attendee. What is the rationale behind testing ceftazidime simultaneously with ceftazidime-avibactam?
I don't think it needs to be simultaneous. I think you just should test both, and in most cases ceftazidime will have been tested in the first screen. So first line antibiotics will be tested in the lab by disc testing usually, and then ceftazidime-avibactam will be tested later on if the patient is not doing well and if it looks as if Cazavi will be useful. The reason I say you need to have tested them both, not necessarily simultaneously, is the fact that if you get an eye result with the ceftazidime, and an S result with the Ceftazidime-avibactam, then you just need to be aware that you're not giving out spurious information. So where ceftazidime will do and Ceftazidime-avibactam, you don't really need to use that in those particular cases. Yeah, I think that's probably the reason. Not necessarily simultaneous, but I think you need to test both.
Yeah, that makes sense. Okay, so next question is from Maria Damala, and she asked, for de-repressed AMP-C antibacteriales, both Ceftazidime-avibactam and Ceftolozane-tazobactam have the same activity?
I think in the Livermore paper that you were on, Katie, they had, I think the activity was up in the 90s, wasn't it, for both?
I think so. I mean, we test both agents standardly on our gram-negative panel. I would say probably with Enterobacter, for example, we probably see more Ceftolozane-tazobactam resistance than we do Ceftazidime-avibactam resistance. So I think that there probably is a bit of a difference, but yeah,
I think
still pretty good.
Generally people go with Ceftazidime-avibactam for enterobacteriales anyway. I think that's just at the top of the list when you're choosing those agents, the newer agents. You know, Ceftolozane-tazobactam is almost always spoken about in terms of treating Pseudomonas infections. So I maybe there is a little difference, but I think generally clinically that's how they're used.
Yeah, that would be my interpretation as well. Generally, most of the conversations we have in the reference lab here for Ceftolozane-tazobactam are more related to Pseudomonas than the antibacterials.
Yeah.
Okay, so the next question we have, I think, might help if we kind of broke it down as two pieces because it's quite a general question. So again, from an anonymous attendee, and they've said phenotypic testing seems to be better than genotypic detection of antimicrobial resistance. Please comment. So I wonder if we need to maybe break it down as in a kind of general comment and then try and address it with specific relevance to the agents you've discussed today.
Yeah. How long have you got? This is quite a difficult question to answer, to be honest. I could give you a very short answer, and I could say, yes, phenotypic testing is better for a number of reasons. For generally, it tells you what the agents are susceptible, rather than resistant. So you know, you get an immediate response within 24 hours, whereas WGS takes 3 days at the most, at the very least, to get an answer. So it's more responsive. Phenotypic testing is cheaper as well, although that's probably not a factor, but it is in most big labs. And there are genes and there are resistance mechanisms out there that we don't know about yet. So take the drugs that we've just been talking about. We don't know what the resistance mechanisms are. They may be multifactorial, and they may not be picked up with the current databases as well. There may be some resistance. So for example, with carbapenemases, there is a multitude of alleles for each carbapenemase gene. And if it's new and it's not in the database, it won't be picked up. There are some really nice algorithms that have been written now to look at different resistance mechanisms and combining not just the gene, the acquired resistance mechanisms like carbapenemases, the SBLs and such like. So there is a lot of those resistance mechanisms, such as phenotypical or physiological changes, so mutations in PVPs and all that kind of stuff, which generally the databases don't pick up because they're not acquired. But there are some algorithms that are joining all of the resistance mechanisms together now to put into a vast sort of resistance mechanism detection database. But it's quite difficult to do that, and you know, different people will use different databases. So we're then going to be back into a situation where we can pick it up in Wales, but we can't pick it up in Leicester, and we can pick it up in Sweden, but we can't pick it up in France, you know, because we're using different databases. So whereas the DIS test, the EUCAST DIS test, covers all of those areas and will give you a standardized method and a standardized result. So generally, I think it is, for lots of different reasons. In particular, with these newer agents, it's difficult for whole genome sequencing to detect the resistance because we don't know what it is.
I think you've covered pretty much everything there, Mandy, in quite a short answer. Yeah, I
think I could have gone on for a while.
I know, I was just thinking about it myself, and there's so many different areas to consider. I mean, whole genome sequencing, yeah, has the potential, but I don't really think at the moment it's there yet, certainly for the organisms that you're talking about today. Clearly for some of the other organisms, as a more kind of looking at it in a more general context, there are some organisms where you can quite accurately predict resistance. And I think sort of from the reference lab side here at Collindale, there are some reference services that are moving more towards genotypic prediction.
Well, TB, you know, TB is for whole genome sequencing is great for TB because TB is difficult to grow, difficult susceptibility test. And the resistance mechanisms to the agents, there's only a few agents to start off with. There's only one bug. So it's quite easy to detect those genes. And that's, so it's easy for that. And that's great, you know.
Yeah, but still a lot to unpick, unfortunately, for the gram-negatives, as always is the case.
Yes.
Okay.
Many of them, isn't there?
Yeah. Okay, so I've got a few questions if you don't mind answering those as well.
Yeah, that's fine.
Okay, where shall I start? So I mean, I was just interested really to hear sort of your experience from your reference lab. So presumably you're testing these agents that you've spoke about today. I mean, do you see much resistance, and do you kind of look at the underlying resistance mechanisms?
We do see a little bit of resistance to Ceftolozane in Pseudomonas. That's mainly where my lab tests the newer agents. That's the most frequent one we test because we have a large CF unit here in Cardiff, so we get a lot of Pseudomonas through that we test, and you know, we cover the whole of Wales for CF, so you know, we get that more often than not. We don't tend to look at the resistance, but we do have a study ongoing with St George's where we are looking at whole genome, well, we've whole genome sequenced quite a lot of Pseudomonas aeruginosa from CF and from non-CF. Look at the anything that's associated with Ceftolozane resistance, and the isolates have come from all over Britain. So that's ongoing. We've got an ECMID poster in ECMID this year, but I don't think we found anything too enlightening, to be honest. A couple of the AMP-C mutations that other people have noted, particular efflux pumps, again, it's just an association study, so we haven't done any in-depth research into it. We're just correlating resistance with presence of genes and mutations. So we haven't found anything different to what anybody else has found and what I spoke about earlier on, really.
Okay.
With Cas-Avi, we don't tend to have a great deal of carbapenem resistance in Enterobacterialis in Wales. I have tested Cas-Avi quite a lot at the moment, and one of the reasons I put in the synergy testing with that as Trianam was because that's the one thing that we do quite often if we've got a metallobutyllactamase organism infection, and we tend to do that just for the clinicians' benefits, just to give them a bit of confidence.
Yeah. Do you ever see many isolates where you've done that Ceftaz-Avi as Trianam synergy testing where you don't actually see that there's synergy and you'd expect that there would be?
Not at the moment, no. Even slightly, you get a slight bit of synergy. We haven't seen anywhere there's just been none at all. But then we do tend to get the bog standard NDM E. coli, so you don't get anything truly weird and wonderful.
Yeah. Yeah, I mean, just to add from the sort of Collindale perspective, thankfully most of our Ceftazidime-avibactam testing, where we'd expect to see susceptibility, we do. We did have one patient a couple of years ago where we had one of these mutant KPCs in, which was quite interesting. Really nice kind of classic change of the phenotype as well, from the carbapenemase to the more ESBL-like and the sort of reduced resistance to meropenem. So that was quite exciting for us.
Cool.
Okay. So at the moment, with the NEQAS distributions that are sent out, the participants aren't asked to look for susceptibility to the agents that you've mentioned today. Do you know if there's any plans that they might add these antibiotics to those antibiotic panels we're asked to test?
So funny you should say that. Yeah, I mean, I think at the moment, not many people are testing. You know, one of the reasons I did the talk today was to sort of highlight some of the drugs that people may be testing either this year or next year. You know, the prevalence of, you know, MDR bugs are not going to go away. You know, it's not going to go away. They're just going to get more and more of these through the main labs, and so they will be testing these agents in future. We did, at a recent NEQAS meeting, we did discuss adding them, and we decided against it for next year, but we are thinking of adding the odd one down the line. And I think that's right. You know, we probably won't ask people to test a ribociclib. I mean, to be honest, we ask people to test what they test, generally standard. And I don't think any of these drugs will be standard. I think most of them will only be pulled out the drawer when we need them. But I know from later on this year, Wales will be testing Ceftazidime-avibactam and Ceftolozane-tazobactam as standard. On the first line.
Okay. And the last question I had, really, so for labs that might just be testing Cefiderocol susceptibility by disc diffusion only, is there sort of any guidance as to how they might interpret their zone diameters if there are no EUCAST breakpoints? Because I know sometimes we do get kind of clinicians ask for susceptibility testing for Cefiderocol against, you know, some sort of weird CF bugs. Is there any advice you can give for those labs, or is it really a case of you have to determine the MIC?
I think you're going to have to determine the MIC. I think that's standard for anything, any bug or drug without a clinical breakpoint. And obviously, if it's an MIC, you've got the PK/PD, and that's the only thing you've got to do an MIC. As you saw with the Cenotrophomonas and the Acinetobacter, although the disc diffusion, you can do it, and the note suggests that you've got a zone diameter criteria that you can use, which relate to the PK/PD. They're different for different bugs. So it was 17 and 20. So the disc diffusion doesn't always tally. So yeah, I think you're going to have to do an MIC.
Okay.
As tricky as it is.
Yeah. Okay, well, that's it with the questions that we've received for today then. So thanks very much, Mandy.
Thank you, Katie. It was great.
Thanks.